Welcome, everyone, to the Deep Dive. We are absolutely thrilled you are joining us today.
Truly thrilled. We have a massive topic today.
We really do. And whether you are a dedicated learner, maybe you're a student intensely prepping for a major biology exam like the Neet, or simply someone who is insanely curious about the fundamental engine of life on Earth, you are in the exact right place.
Yeah, because we are going to be looking at something so ubiquitous, we often take it completely for granted.
You look out the window, you see green leaves, and you probably don't think twice about them.
Right, but what is happening inside those leaves is arguably the most important chemical reaction happening on our planet at any given second.
It really is the bedrock of almost all life.
We are talking about photosynthesis in higher plants.
But, and this is key, we are going far beyond the standard elementary school definition of, plants using sunlight to make food.
Oh, way beyond. We are going to deconstruct the machinery at a molecular level.
The goal here is to really understand the architecture, the chemistry, and the sheer evolutionary ingenuity of this process.
Exactly. We want to understand the exact mechanics of how light is captured and transformed into physical, usable chemical currency.
And we have a very specific mission for you today. We are going to conduct a meticulous accountant-style audit of photosynthesis.
I love that framing. An audit.
Right. We are going to treat the plant cell like a highly optimized manufacturing plant with a strict balance sheet.
So we'll track the exact stoichiometry, the precise molecular accounting of both the light-dependent and the light-independent reactions.
We're going to break down the exact income and expenditure of the cellular energy currencies.
That's the ATP and NADPH.
We'll explore the strict division of labor inside the plant's cellular factories, the chloroplasts.
And maybe my favorite part, we are going to uncover a brilliant evolutionary tax loophole.
Yes. The Krantz anatomy.
Right? Which is used by a special group of plants to avoid massive biological inefficiency.
Treating this biological process like an economic system is actually the perfect way to visualize it.
Because knowledge is most valuable when understood and applied. When you frame photosynthesis as a factory floor with raw materials, energy currencies, assembly lines, and waste products, the abstract chemistry suddenly becomes incredibly intuitive.
You could just see it.
Yeah. We are going to follow the flow of energy, the movement of individual electrons, and do some meticulous counting of carbon atoms.
Okay, let's unpack this. Let's actually start by looking at how humanity even figured out what was happening inside a green leaf.
Because for the longest time, the inner workings of plants were a complete black box.
People just thought plants ate dirt.
Literally, yeah. They thought the mass came from the soil.
So we need to look at the early auditors of this process, the scientists who first started putting the pieces of the puzzle together, because their methods were brilliantly simple.
It is wild to think about how little we knew just a few centuries ago. If we go back to the 1770s, a scientist named Joseph Priestley performed a series of experiments that fundamentally changed our understanding of the relationship between plants and the air we breathe.
This is the same Priestley who is generally credited with discovering oxygen a few years later.
The very same. He designed an experiment that is now considered a classic, using a simple glass bell jar.
The classic bell jar setup. If you are prepping for an exam, visualizing this specific experiment is usually a must.
Absolutely. So Priestley takes a completely sealed glass space, the bell jar, and puts a burning candle inside.
What happens?
The candle quickly burns out.
Right.
Then he puts a live mouse in the bell jar.
And the mouse soon suffocates.
Exactly. So his initial conclusion is incredibly logical for the time. He deduces that both a burning candle and a breathing animal somehow damage or deplete the air.
They use up some invisible vital quality.
But the breakthrough comes with his next step. Priestley places a living mint plant inside that exact same sealed bell jar, right alongside the mouse and the candle.
And the result was completely counterintuitive to the scientific understanding of the era.
The mouse survives. And if he attempts to light the candle, it continues to burn.
From this, Priestley formed a revolutionary hypothesis. Plants somehow restore to the air whatever breathing animals and burning candles remove.
It's elegant.
Though whenever I think about this, I always wonder about the physical logistics. Like, how did Priestley actually light the candle inside a sealed glass jar days later without lifting the lid and ruining the airtight seal?
That is a great question. It turns out he used a magnifying glass.
A magnifying glass?
Yeah, to focus sunlight onto the candle wick from the outside to rekindle it.
That is amazing. It makes you appreciate the incredible practical ingenuity these early researchers had to employ. You can't just use modern sensors. You have to invent the methodology from scratch.
That is a phenomenal detail, and it highlights the physical reality of historical science.
Building on Priestley's work, a Dutch scientist named Jan Ingenhousz took that exact bell jar setup but introduced a vital new variable to the equation.
Light?
He placed the entire setup in total darkness and then separately in bright sunlight.
And Ingenhousz definitively showed that sunlight is absolutely essential to this plant process that purifies the air.
He also took things a step further with an elegant experiment using a submerged aquatic plant.
I love the aquatic plant experiment because it provides such immediate visual feedback.
Exactly. When he placed this aquatic plant in bright sunlight, he observed tiny bubbles forming all around the green parts of the plant.
But when he moved the setup into the dark, the bubbles stopped forming entirely.
He later managed to collect and identify the gas in these bubbles as oxygen.
Which proved two crucial things. First, that it is specifically the green parts of the plants that release oxygen, and second, that they only do so in the presence of sunlight.
So piece by piece, the factory is being understood. We jump forward to the mid-19th century with Julius von Sachs.
He provided the first real evidence that plants actually produce a physical product, glucose, when they grow, and that they tend to store this glucose as starch.
He also pinpointed that the green substance, what we now call chlorophyll, isn't just floating freely in the plant cells.
Right. It is locked inside highly specific specialized bodies, which we now recognize as chloroplasts.
And that brings us to one of the most clever experiments in early biophysics, conducted by T.W. Engelmann.
He wanted to understand which specific colors of light were most effective at driving photosynthesis.
So he used a glass prism.
He split white light into its individual spectral components, essentially creating a miniature rainbow.
He then shined this split light onto a strand of green alga called Cladophora.
But his method for detecting the rate of photosynthesis is what makes this so brilliant. He didn't have chemical assays, he used biology.
He placed the algae in a suspension of aerobic bacteria.
Wait, aerobic meaning bacteria that actively seek out and require oxygen to survive?
So he basically used a swarm of microscopic bacteria as living, moving oxygen sensors.
Precisely. He watched under a microscope to see where the bacteria congregated.
And he observed that they swarmed massively in the regions illuminated by blue and red light.
By tracking the bacteria, he essentially mapped out the very first action spectrum of photosynthesis.
He proved that blue and red light are the primary drivers of the process.
And as we now know, this roughly mirrors the absorption spectra of the chlorophyll pigments themselves.
By this point, the basic empirical equation of photosynthesis was coming into focus. Scientists knew the inputs were carbon dioxide and water, and the outputs were carbohydrates and oxygen, all driven by light.
So the old-school textbook equation was written as CO2 plus H2O yields CH2O, representing a basic carbohydrate plus oxygen.
But embedded in that seemingly simple equation was a massive, fundamental misunderstanding, right?
A very profound misunderstanding. For decades, the global scientific consensus was that the oxygen released by plants originated from the carbon dioxide.
The assumption was that the plant took in CO2, literally split the carbon away from the oxygen, kept the carbon to build its sugars, and simply exhaled the leftover oxygen gas.
It makes intuitive sense, but it was completely wrong. It took a microbiologist named Cornelius van Niel to shatter that assumption.
How does a microbiologist studying bacteria end up rewriting the fundamental rulebook for plant biology?
By stepping back and looking at the wider tree of life, van Niel was studying organisms that photosynthesize but do it a little differently. Specifically, purple and green sulfur bacteria.
Okay, sulfur bacteria.
Now, these bacteria use light to build sugars from CO2, just like plants. But they do not use water, H2O, as their hydrogen source. Instead, they live in environments rich in hydrogen sulfide, H2S.
Okay, so they use hydrogen sulfide instead of water.
Right. And here is the kicker. When these sulfur bacteria photosynthesize, they do not release oxygen. Instead, they release elemental sulfur or sulfate as a waste product.
Van Niel looked at this and realized something profound.
Exactly. If using H2S results in sulfur being released, then the oxygen released by normal green plants must be coming from their hydrogen source, water, H2O.
That is a massive paradigm shift, because if the oxygen comes from the water, that means the plant is actively tearing apart water molecules, which requires an immense amount of energy.
Exactly. Van Niel deduced that photosynthesis is essentially a light-dependent redox reaction.
A suitable oxidizable compound, in the case of plants, water, donates hydrogen to reduce carbon dioxide into carbohydrates.
Therefore, the true overall balanced equation for photosynthesis in higher plants is 6 molecules of carbon dioxide plus 12 molecules of water in the presence of light energy yields 1 molecule of glucose plus 6 molecules of water plus 6 molecules of oxygen.
And for anyone who loves an airtight balance sheet, that equation is incredibly satisfying. All 6 molecules of O2 released into the atmosphere come entirely and exclusively from the 12 molecules of water on the input side.
But this equation isn't a single magical event. It's a summary of a wildly complex, multi-step manufacturing process.
So let's transition from the history and actually walk onto the factory floor. Let's look at the physical location where all this happens.
We know photosynthesis occurs primarily in green leaves, specifically within the mesophyll cells, which are absolutely packed with chloroplasts.
Interestingly, these chloroplasts aren't static. They actively move around inside the cell cytoplasm to optimize their light exposure.
If the light is optimal, they align their flat surfaces parallel to the cell walls to catch maximum rays.
If the light is too intense and potentially damaging, they turn perpendicular to minimize exposure.
But the real magic happens when we look inside the chloroplast itself.
If we look at an electron micrograph, it isn't just a chaotic soup of chemicals. There is a very strict architectural division of labor. It looks like a highly organized industrial park.
The spatial accounting here is vital. You basically have 2 distinct operational zones. First, you have a complex, highly folded membranous system.
This consists of the grana, which look like stacks of coins, each coin being a thylakoid,
And the stroma lamellae, which are flat membranous tubules that connect the different grana stacks together.
This entire intricate membrane network is the power generation plant of the cell.
So the membrane is where the physical trapping of light energy actually occurs.
The membrane system houses the pigments that catch the light, and it uses that solar energy to directly synthesize our 2 forms of chemical currency.
ATP, which is the raw energy, and NADPH, which provides the reducing power.
Because these reactions absolutely require the direct input of light energy, they are called the light reactions or the photochemical reactions.
Okay, so that's the power plant generating the cash. Then, surrounding this entire membranous network is a fluid-filled matrix called the stroma.
If the membranes are the power plant, the stroma is the assembly line.
Exactly. The stroma contains a complex suite of enzymes. These enzymes take the carbon dioxide pulled from the air and use the ATP and NADPH generated by the power plant to synthesize sugars.
Because these specific enzymatic reactions do not directly absorb light photons to function, they have traditionally been called the dark reactions or the carbon reactions.
We really need to hit pause here and clarify something, because the terminology dark reactions is probably one of the most confusing misnomers in all of biology. It trips up learners constantly.
It is a terrible name, historically speaking. Dark reactions implies that they happen at night or that they require darkness to function.
That is completely false. The dark reactions happen almost entirely during the day.
The term simply means they are not directly light-driven.
But they are completely dependent on the products of the light reactions. If you pull the plug on the power plant, the assembly line stops immediately.
Precisely. If you place a plant in sudden total darkness, the light reactions stop instantly. The dark reactions will actually continue to run for a few milliseconds or seconds.
Just long enough to burn through whatever leftover ATP and NADPH currency is floating around in the stroma.
Once that currency is depleted, the dark reactions grind to an absolute halt.
Turn the lights back on, currency is minted again, and the sugar assembly line starts back up. So they are entirely light dependent, just indirectly.
Okay, we have our 2 departments clearly defined. The membranous power plant handling the light reactions and the stroma assembly line handling the dark reactions.
Let's zoom all the way in on the power plant first. We need to understand exactly how revenue is generated. We need to look at the workforce, the plant pigments.
If you do a chromatographic separation of the pigments in a standard green leaf, you quickly realize it's not just one uniform green substance doing all the work.
There's a whole team of pigments, 4 main ones, to be exact.
You have chlorophyll A, which usually shows up as a bright or blue-green band on the chromatogram.
You have chlorophyll B, which is more of a yellow-green.
Then you have the xanthophylls, which are yellow, and the carotenoids, which range from yellow to yellow-orange.
Now, if we look back at Engelmann's action spectrum, where the bacteria swarmed the blue and red light, and we overlay the absorption spectrum of chlorophyll A, the two graphs line up almost perfectly.
Chlorophyll A absorbs red and blue light incredibly well, and that is exactly where photosynthesis peaks.
This proves that chlorophyll A is the chief worker. It's the primary pigment responsible for trapping light energy.
But if you look really closely at those graphs, there isn't a perfect 100% overlap.
Photosynthesis is still happening, albeit at a lower rate, in the wavelengths of light that chlorophyll is really bad at absorbing.
How is that possible if chlorophyll is doing the heavy lifting?
Because chlorophyll A doesn't work alone. It acts as the manager, but it has a massive support staff made up of those other pigments.
Chlorophyll B, the xanthophylls, and the carotenoids.
We call these the accessory pigments. They have two absolutely vital jobs. First, they act as a broader antenna system.
They absorb light at the wavelengths that chlorophyll emisses, and they physically transfer that captured energy over to the chlorophyll molecules.
This significantly widens the spectrum of light the plant can profitably utilize.
So they are essentially funneling extra energy to the boss. What is their second job?
The second job is pure protection. The incoming solar radiation is incredibly intense. If chlorophyll A were to absorb all that energy completely unshielded, it would literally undergo photooxidation.
It would be destroyed by the very energy it's trying to harvest.
The accessory pigments act like a molecular sunscreen, absorbing excess energy and safely dissipating it to protect the vital chlorophyll at the center of the operation.
Okay, so we have our pigment workforce defined. Now we need to see how they actually manufacture the currency. This is the photochemical phase.
It involves light absorption, splitting water, releasing oxygen, and forming our high-energy intermediates, ATP and NADPH.
And to do this, the pigments are organized into highly structured machines.
They are. The pigments are not just floating randomly in the thylakoid membrane. They are organized into two discrete, massive protein complexes called light harvesting complexes, or LHCs.
These LHCs are the core components of two larger functional units called Photosystem I and Photosystem II.
I want to throw out a quick warning for anyone taking a test on this. The names, Photosystem I and Photosystem II, are named strictly in the historical sequence of their discovery, not in the sequence in which they actually function during the light reaction.
It's a classic trap. Photosystem II is actually the first stop on the assembly line.
A very important distinction. Now, each of these photosystems has an antenna complex made up of hundreds of those accessory pigment molecules we just talked about.
They act like a giant satellite dish, catching photons of light and funneling all of that energy to a single, highly specific molecule of chlorophyll A that sits at the very center.
This central molecule is called the reaction center.
And the reaction centers are slightly different in each photosystem, right? They are tuned to different frequencies.
Yes. The reaction center in photosystem is a chlorophyll, a molecule that has its absolute peak absorption at 700 nanometers. Because of this, it is referred to as P700.
In photosystem 2, the reaction center chlorophyll A peaks at 680 nanometers, so it is known as P680.
Okay, here is where we need to trace the physical movement of the electrons. This process is called non-cyclic photophosphorylation, but it's more commonly known as the Z-scheme.
Let's follow an electron on this journey, starting right at photosystem 2, or P680.
Sunlight hits the antenna pigments of photosystem 2. The energy from those photons is funneled directly into the P680 reaction center.
When P680 absorbs a photon of red light at that 680 nanometer wavelength, the energy is transferred to one of its electrons.
This electron gets so energized, so excited, that it literally breaks free from the atomic nucleus.
It performs a quantum leap into an outer orbit and is ejected from the chlorophyll molecule entirely.
So the light physically knocks an electron out of the pigment.
Exactly, but it doesn't just fly off into the void. It is immediately caught by a primary electron acceptor molecule waiting right there in the membrane.
This acceptor molecule then passes the highly energized electron into an electron transport system.
Now, I hear the phrase electron transport system, and I imagine tiny molecular wires. But what does this actually consist of?
It consists of a series of complex proteins, primarily cytochromes. You can think of them like a bucket brigade or staircase.
As the electron is passed from one cytochrome protein to the next, it is moving downhill in terms of its oxidation reduction or redox potential.
Let me stop you there, because redox potential can sound intimidating if I'm visualizing this. Is redox potential basically like gravity for electrons?
That is an excellent way to think about it. Imagine a ball at the top of a staircase. It has high potential energy.
As it bounces down each step, it moves to a state of lower energy, a more stable state, and it releases energy with each drop.
In the electron transport chain, each cytochrome protein has a slightly stronger affinity for the electron than the one before it.
So the electron naturally flows down the chain from lower affinity to higher affinity, releasing a little bit of its stored energy at each transfer.
Okay, so the electron rolls down the cytochrome staircase, releasing energy as it goes. Where does it eventually land?
It lands at the next major station on the factory floor, Photosystem 1.
Now, while Photosystem 2 is busy ejecting an electron, Photosystem I has also been absorbing light.
Its reaction center, P700, gets excited by light at 700 nanometers, and it too ejects an electron out of its orbit.
So the electron that just rolled down the staircase from Photosystem 2 essentially falls right into the empty hole left behind in Photosystem 1.
It's a perfect relay system. But now Photosystem I has ejected a newly excited electron. Where does that one go?
That electron is passed to a different primary acceptor, one with an even higher redox potential.
And then it is moved downhill once again through a shorter transport chain.
But this time, the final destination is not another photosystem. The final destination is a molecule called NADP plus May.
The addition of these two energetic electrons, along with some protons from the surrounding fluid, chemically reduces NADP plus into NADPH plus H plus May.
And there it is. We have just successfully minted one of our major cellular currencies.
So if we step back and trace this entire path on a graph of redox potential, putting energy level on the y-axis, it looks exactly like a letter Z tipped on its side.
You start at PS2, spike straight up to an acceptor, slant downwards through the cytochromes to PSI, spike straight up again to the next acceptor, and slant down to NADP plus REA, the famous Z scheme.
It's a beautifully choreographed flow of energy. But as an accountant looking at this, you should immediately spot a glaring deficit in the balance sheet.
Right. If Photosystem 2 is constantly getting hit by light and constantly ejecting electrons to send down the chain,
It's going to run out of electrons almost immediately.
how does it stay in business?
It needs a constant source of raw capital to replace those lost electrons. And for a plant, that capital comes from splitting water.
This is an incredible chemical feat.
Photosystem 2 is physically associated with a water-splitting complex. This complex takes liquid water and literally rips it apart into three distinct components.
Protons, which are just hydrogen ions, oxygen atoms, and electrons.
The exact equation is 2 molecules of H2O are split to yield 4 protons, 1 molecule of O2 gas, and 4 electrons.
Those 4 freshly liberated electrons are fed directly into the P680 reaction center of Photosystem 2, perfectly replacing the ones that were just ejected down the Z scheme.
The oxygen atoms immediately combine to form O2 gas, which diffuses out of the leaf and becomes the oxygen we breathe.
But the physical location of this water splitting is vital to the next step of our audit. The water splitting complex is located exclusively on the inner side of the thylakoid membrane.
Exactly. So when the water is split, the oxygen escapes, but those protons, the H+ ions, are trapped.
They are released directly into the lumen, the enclosed inner space of the thylakoid sac.
This localized dumping of protons into a confined space is going to be incredibly important for synthesizing ATP.
Now, before we get to ATP, there is an alternative pathway we need to discuss. We just meticulously tracked the non-cyclic Z scheme, which produces both NADPH and ATP.
But sometimes the plant uses cyclic photophosphorylation. Why and how does that work?
To understand the how, we have to look back at our architectural map of the chloroplast.
Remember, the membrane system has 2 parts: the grana, the dense stacks, and the stromal-lamellae, the flat tubes connecting the stacks.
The membranes of the grana are fully equipped. They possess both Photosystem I and Photosystem II.
But the membranes of the stromal-lamellae are fundamentally different. They lack Photosystem II entirely.
Furthermore, they lack the NADP reductase enzyme, which is the machine needed to finalize the production of NADPH.
So the stromal-lamellae are essentially operating with half a factory. They only have Photosystem I.
Precisely. So what happens when light hits these specific regions?
P700 absorbs the light, gets excited, and ejects an electron to its primary acceptor.
But because there is no NADP reductase enzyme waiting at the end of the line to catch it, the electron has nowhere to go. It reaches a dead end.
So it gets shunted backward. It is passed back into the cytochrome electron transport chain and simply cycles back down to the P700 reaction center it just left.
A closed loop, a cyclic flow.
Yes, and because the electron is still rolling down that cytochrome staircase, it is still releasing energy.
That energy is used to drive the synthesis of ATP.
But because it's a closed cycle, several things don't happen. No water is split because PS2 isn't involved.
No oxygen is released.
And crucially, no NADPH is synthesized. The cyclic flow results only in the synthesis of ATP.
We're going to see later exactly why the plant needs this weird one-sided cyclic pathway. It's a brilliant accountant's trick to balance the final budget.
But first, we need to address the elephant in the room. We keep saying the energy from the falling electrons is used to synthesize ATP.
But what is the actual physical mechanism? How do you turn the kinetic energy of a tumbling electron into a tangible chemical bond?
This is one of the most elegant mechanisms in all of biology, described by the chemiosmotic hypothesis.
If we stick to our analogies, we are moving from the factory floor to a hydroelectric dam.
The core principle of chemiosmosis is that the synthesis of ATP is directly linked to the creation of a proton gradient across a membrane.
So the plant acts like an engineer, building a dam and packing a massive reservoir of water, in this case protons, behind it.
Let's do a strict audit of how the plant builds this gradient. The goal is to pack the inside of the thylakoid, the lumen, with protons, while draining them from the outside, the stroma.
There are 3 distinct mechanisms driving this. What is reason number 1?
We already covered reason number 1, the water splitting complex. Because water is split exclusively on the inner side of the membrane, every single time a water molecule is ripped apart to provide electrons, the resulting protons are dumped directly into the lumen reservoir.
That is a constant, steady deposit of protons into the vault.
Okay, direct deposit via water splitting. What is mechanism number 2?
Mechanism number 2 is an active pumping system, and it's tied directly to our electron transport chain.
Remember that as electrons move from photosystem 2 to photosystem I, they pass through primary acceptors.
The primary acceptor for PS2 is located towards the outer side of the thylakoid membrane, facing the stroma.
Right, it catches the ejected electron.
But here's the trick. When it passes that electron to the next stage, it doesn't pass it to a standard electron carrier. It passes it to a hydrogen carrier.
To become a hydrogen atom, this carrier molecule requires both an electron and a proton.
So it grabs the electron from the chain, but to balance its charge, it has to literally reach out and snatch a proton out of the surrounding stroma fluid.
Exactly. It pulls a proton from the stroma, effectively lowering the stroma's proton concentration.
Then this carrier physically pivots or moves across the membrane to the inner side.
When it passes the electron back into the cytochrome chain, it can no longer hold onto the proton.
So it releases the proton, but it releases it on the inside, into the lumen.
That is incredibly clever. The electron transport chain acts as an active physical pump?
The energy of the electron rolling down the staircase is essentially used to power a turnstile that grabs a proton from the outside and shoves it to the inside.
It is a massive transfer of wealth.
And the final blow to the stroma's proton concentration is mechanism number 3, the NADP reductase enzyme itself.
This enzyme is located on the stroma side of the membrane. Its entire job is to reduce NADP plus into NADPH.
To do this, it requires the electrons arriving from photosystem I, but it also requires protons. Where does it get them?
It sucks them straight out of the stroma fluid.
So let's review the ledger. We have a 3-pronged attack, creating a massive imbalance. Water splitting actively adds protons to the inside.
The electron transport turnstile actively moves protons from the outside to the inside.
And the NADP reductase enzyme actively consumes the few protons left on the outside.
The result: the stroma is entirely depleted of protons, while the lumen becomes highly acidic and absolutely packed with them. We have built a massive electrochemical gradient across that thylakoid membrane.
The reservoir behind the dam is full. There's immense potential energy stored in that gradient.
The protons are packed so tightly and the charge difference is so great that they desperately want to diffuse back out into the stroma to balance the concentration.
But the lipid membrane is completely impermeable to them. They are trapped.
So how does the plan cash out this potential energy? How does it open the floodgates?
It provides exactly one highly controlled escape route, a massive transmembrane enzyme complex called ATP synthase.
This is our turbine. The ATP synthase enzyme consists of 2 distinct physical components.
The first part is called CF0. It is embedded directly within the thylakoid membrane and forms a physical channel.
This channel allows the facilitated diffusion of protons to flow across the membrane, moving from the high concentration in the lumen back to the low concentration in the stroma.
The floodgates open, and the water rushes through the dam.
Exactly. And the second part of the enzyme is called CF1. This portion protrudes on the outer surface of the thylakoid membrane, sticking out like a knob into the stroma.
As the massive stream of protons rushes through the CF0 channel, the sheer physical kinetic energy of that movement causes a structural conformational change in the CF1 particle.
It literally forces the protein to change shape.
The turbine physically spins.
It acts just like a microscopic rotary motor, and this physical shape change provides the exact amount of activation energy needed to catalyze the chemical bond formation between ADP, adenosine diphosphate, and inorganic phosphate, forging them into the high-energy ATP molecule.
The breakdown of the physical gradient is the force that churns out the chemical currency.
It is breathtaking to visualize, and just like that, the light reactions are complete.
The power plant has done its job. The plant has successfully utilized sunlight and water to produce a surplus of oxygen, which diffuses away, and a massive vault full of chemical currency.
ATP and NADPH, which are released directly into the stroma.
Which is the perfect location, because the stroma is where our next phase begins. We are moving from the power plant to the assembly line.
The plant now has a massive bank account of ATP and NADPH. Now it is time to spend them to build actual biomass.
We are entering the dark reactions, specifically the Calvin cycle, also known as the C3 pathway.
The detective work to uncover this pathway is a great story in itself. We know it requires taking CO2 and fixing it into a solid form, but how did we trace the exact steps?
This is a classic example of how technological advancements drive biological discovery. Right after World War II, researchers suddenly had access to a powerful new tool, radioactive isotopes, specifically carbon-14.
A scientist named Melvin Calvin used this radioactive carbon to track the exact path of carbon fixation in algal photosynthesis.
By feeding the algae radioactive CO2 and then flash freezing them to stop the chemical reactions at split-second intervals, he was able to trace exactly where the radioactive carbon atoms went.
He essentially put a tiny GPS tracker on the carbon atom. And his biggest initial discovery was identifying the very first stable product of this fixation process.
What did it turn out to be?
It was a 3-carbon organic acid called 3-phosphoglyceric acid, or PGA for short.
Because this very first stable intermediate has exactly 3 carbon atoms, this entire sequence is known as the C3 pathway.
Now, here's where the scientists got initially tripped up by their own logical assumptions. They knew the first stable product was a 3-carbon molecule, PGA.
And they knew they were adding 1 carbon atom from the atmospheric CO2.
So they applied basic elementary school math. They assumed if we were adding 1 carbon to an unknown acceptor molecule and the result is a 3-carbon product, then the acceptor molecule absolutely must be a 2-carbon compound.
1 plus 2 equals 3.
It seems like an airtight logical deduction, and researchers spent years scouring the chloroplast trying to isolate this elusive 2-carbon acceptor.
But the actual biochemistry yielded a completely unexpected result. The primary acceptor molecule wasn't a 2-carbon compound at all.
It was a 5-carbon ketose sugar called ribulose bisphosphate, or RuBP.
Wait, hold on. Let's run that math. You have a 5-carbon acceptor, RuBP, plus 1 carbon from CO2. That equals 6 carbons. But the product they found, PGA, only has 3 carbons. That balance sheet doesn't work.
Ah, but it does, just in a way they didn't expect. The 6-carbon intermediate that forms when CO2 binds to RuBP is incredibly unstable.
It is so unstable that it instantly, almost simultaneously, cleaves perfectly in half.
It splits into 2 identical molecules of 3-carbon PGA. So 5 carbons plus 1 carbon equals 2 times 3 carbons.
The stoichiometry balances perfectly. The mechanism was just a surprise.
I love that. Biology doing math its own way. So Melvin Calvin and his team work out the whole pathway and realize it operates in a continuous loop.
The starting material, RuBP, has to be constantly regenerated to keep accepting new CO2, hence the Calvin cycle.
To make this digestible for anyone studying the process, we need to break the Calvin cycle down into 3 distinct manageable stages. Let's audit stage 1, carboxylation.
Carboxylation is arguably the most crucial step of the entire cycle.
This is the moment of actual carbon fixation, where inorganic CO2 from the atmosphere is finally pinned down into a stable organic intermediate.
The CO2 is chemically combined with our 5-carbon acceptor, RuBP. This reaction is entirely dependent on a specific enzyme to catalyze it.
And this enzyme is widely considered the most abundant and arguably the most important protein on the face of the Earth, RuBP carboxylase.
But if we look at the specific biochemistry, it has a slightly longer, incredibly important name, RuBP carboxylase oxygenase, or RuBisCO for short. Why add the oxygenase part?
We are going to dive deeply into the oxygenase part in a few minutes because it represents a monumental flaw in the system.
But for this specific stage, carboxylation, RuBisCO is acting purely as a carboxylase.
It grabs the CO2, attaches it to RuBP, the unstable 6-carbon intermediate forms, and immediately splits into 2 molecules of 3-PGA.
Okay, carboxylation is complete. The carbon is captured. But 3-PGA isn't a sugar, it's just an acid. We need to upgrade it.
That brings us to stage 2, reduction. This is where the heavy spending happens on our balance sheet.
Yes. To reduce those 2 molecules of 3-PGA into a high-energy sugar, specifically a triose phosphate that will eventually be combined to form glucose, the plant has to finally open its vault and spend its hard-earned currency.
For every single molecule of CO2 that was just fixed, the reduction stage demands an expenditure of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for the actual chemical reduction.
So, per CO2 molecule captured, our cost is 2 ATP and 2 NADPH. The carbon is now successfully reduced to a triose phosphate.
Some of these triose phosphates will be siphoned out of the cycle to synthesize the glucose, sucrose, or starch the plant actually uses to live.
But we can't drain the system entirely. The assembly line must keep moving. So we enter stage 3, regeneration.
If you want to keep capturing CO2, you must continuously regenerate the primary acceptor molecule, the 5-carbon RuBP.
If you run out of RuBP, the entire Calvin cycle comes to a crashing halt. It would be like a car factory running out of chassis.
But rebuilding this complex 5-carbon sugar from the leftover 3-carbon scraps requires a little bit of extra energy.
The regeneration stage requires the expenditure of 1 additional ATP molecule to phosphorylate the intermediate and finalize the RuBP.
Okay, let's step back and do the final ultimate audit. We want to manufacture 1 single molecule of glucose. We know the formula for glucose is C6H12O6. It requires 6 carbon atoms.
How many complete turns of the Calvin cycle are required to build 1 glucose molecule?
Since each complete turn of the cycle captures and fixes exactly 1 molecule of CO2 and a glucose molecule requires 6 carbons, the assembly line must run through the cycle 6 completely separate times.
6 complete turns. Let's calculate the total molecular cost. In the reduction stage, we spent 2 ATP and 2 NADPH per turn. Over 6 turns, that is 12 APP and 12 NADPH.
In the regeneration stage, we spent 1 extra ATP per turn. Over 6 turns, that is 6 ATP. What does the final balance sheet look like?
To synthesize just 1 molecule of glucose, the Calvin cycle demands a grand total input of 6 CO2 molecules, 18 molecules of ATP, and 12 molecules of NADPH.
The final output is 1 glucose molecule, 18 spent ADPs, and 12 spent NADP plus molecules, which head back to the light reactions to be recharged.
If you are studying this, write those numbers down in big bold letters, 18 ATP and 12 NADPH. Look at that ratio. The dark reactions require significantly more ATP than they do NADPH. It's a 3-to-2 ratio.
And this mathematical discrepancy brings us right back to our earlier question about the power plant. Remember the non-cyclic Z scheme.
It produces ATP and NADPH in roughly equal 1-to-1 amounts.
If the plant only ran the Z scheme, it would very quickly run out of ATP while still having plenty of leftover NADPH sitting around. The Calvin cycle would stall out due to a lack of ATP funding.
And that is exactly why the cyclic photophosphorylation pathway exists in the stromal lamellae.
Exactly. The cyclic flow, which only involves photosystem I, operates solely to generate ATP without making any NADPH.
It functions specifically as a supplementary power generator to make up the exact ATP deficit required by the Calvin cycle.
It ensures that the strict 18-to-12 ratio is met perfectly. It is an absolutely flawless, self-balancing biological accounting system.
When you look at the math, it is breathtaking how beautifully and intricately it all fits together. However, evolution is a tinkerer, not a perfect engineer.
We have audited the ideal scenario, the C3 pathway. But if we dig deeper into the fundamental biology, we are forced to confront a massive, glaring inefficiency in this system.
A catastrophic drain on resources. We need to talk about the wasteful tax of photorespiration.
We mentioned earlier that the primary enzyme driving the Calvin cycle is RuBisCO, the most abundant protein on Earth. But RuBisCO has a dark side, a fatal biochemical flaw.
Remember its full name, ribulose bisphosphate carboxylase oxygenase.
Carboxylase and oxygenase, meaning it can interact with both.
Yes. The active binding site of the RuBisCO enzyme is shaped in a way that is just slightly too ambiguous.
It can bind to carbon dioxide, which is what it's supposed to do, but it can also accidentally bind to oxygen.
It is a classic competitive binding situation. Whether it acts as a carboxylase or an oxygenase depends entirely on the relative concentration of the two gases inside the leaf at any given moment.
Now, RuBisCO does have a much greater natural affinity for CO2 than for O2. If the concentrations are exactly equal, it will choose CO2.
But the Earth's atmosphere isn't equal. It is roughly 21% oxygen and only about 0.04% carbon dioxide. Oxygen outnumbers CO2 massively.
Exactly. Because of this overwhelming atmospheric ratio, in a normal C3 plant, some oxygen inevitably forces its way into the active site of RuBisCO instead of carbon dioxide.
And when oxygen binds, the entire assembly line derails spectacularly.
Instead of the 5-carbon RuBP combining with CO2 to form two neat, highly useful molecules of 3-PGA, the RuBP is literally oxidized, burned.
It is cleaved into one molecule of phosphoglycerate, which has 3 carbons and can somewhat be salvaged, and one molecule of phosphoglycolate, which only has 2 carbons and is incredibly toxic to the plant.
This accidental binding initiates a complex salvage operation known as the photorespiration pathway. And let's audit this pathway because the numbers are grim.
What are the outputs? Does it make any sugar for the plant?
None. There is absolutely zero synthesis of sugars.
Does it mint any cellular currency? Does it generate ATP or NADPH?
No, neither ATP nor NADPH is synthesized. In fact, the reality is much worse. To process that toxic 2-carbon phosphoglycolate, the photorespiratory salvage pathway actively consumes previously earned ATP.
It burns through the plant savings just to clean up the mess.
And what is the final insult to injury at the end of this pathway?
It results in the release of CO2. It literally takes carbon that the plant had previously spent massive amounts of energy to fix and capture, and it exhales it back out into the atmosphere as a waste gas.
It is a massive biological deadweight loss.
It is a punitive tax on the plant's productivity that yields absolutely nothing in return. It burns energy, it wastes carbon, and it stalls growth. It just seems to be a purely wasteful process caused by the enzyme's fundamental lack of specificity.
And this structural flaw creates a severe, life-threatening problem for plants in certain environments. Think about a C3 plant growing in a hot, dry, tropical climate.
As the temperature spikes, the plant starts losing water rapidly through transpiration.
To protect itself from drying out, the plant takes defensive action. It closes its stomata. The microscopic pores on the surface of the leaf.
But the stomata are the only doors to the outside world.
Exactly. When the stomata slams shut, 2 catastrophic things happen simultaneously inside the leaf.
First, fresh CO2 can no longer get in, so the CO2 concentration plummets as the Calvin cycle uses up whatever is left.
Second, the light reactions are still running, still splitting water, and still pumping out oxygen gas.
But with the doors closed, that oxygen can't escape. So the internal O2 concentration skyrockets.
High oxygen, extremely low CO2. That is the exact worst-case scenario for RuBisCO.
It is a disaster. Under those specific conditions, RuBisCO abandons carbon fixation entirely and starts binding almost exclusively to oxygen.
Photorespiration runs rampant. The plant basically stops growing and begins to metabolically cannibalize its own resources just to survive the heat of the day.
Which brings us to one of the most brilliant, complex evolutionary workarounds in all of biology. To survive in these brutal environments, a specific group of plants evolved the ultimate tax loophole.
Let's look at C4 plants and the architectural marvel of Krantz anatomy.
How do plants like maize, sorghum, and sugarcane completely avoid this ruinous photorespiration tax?
They realized they couldn't fix the RuBisCO enzyme itself. The flaw is too deeply baked into its structure.
So instead, they evolved an entirely separate front-end mechanism to protect the RuBisCO from oxygen.
They use a biochemical pathway called the C4 pathway, or the Hatch and Slack pathway. But this isn't just a simple chemical tweak. It required a radical physical redesign of the leaf's internal architecture.
This physical redesign is known as Krantz anatomy.
Yes. Krantz is a German word that translates to wreath or halo, and it perfectly describes the specific arrangement of the cells.
If you take a cross-section of a standard C3 leaf, the mesophyll cells are loosely packed. But if you slice open a C4 leaf, you see something very different.
You have a normal outer layer of mesophyll cells, but deep inside the leaf, tightly surrounding the vascular bundles, the veins of the leaf, there are particularly large, highly specialized cells called bundle sheath cells.
They form several dense layers around the veins, looking exactly like a wreath.
And these bundle sheath cells are basically microscopic armored bunkers. They are structurally reinforced.
They have incredibly thick cell walls that are largely impervious to gaseous exchange, and there are absolutely no intercellular spaces between them. They are sealed tight from the outside air.
That physical airtight seal is the absolute key to the entire strategy. In C4 plants, the standard Calvin cycle, the one that relies on the flawed RuBisCO enzyme, does not occur out in the open mesophyll cells where the oxygen is.
It only occurs deep inside these armored, gas-tight bundle sheath cells. RuBisCO is locked in the bunker.
But wait, if the bundle sheath is impervious to gases, how does the carbon dioxide from the air ever reach the RuBisCO inside the bunker to be fixed?
That is where the genius of the Hatch and Slack biochemical pathway comes in. C4 plants have essentially created a spiralized, highly organized molecular smuggling operation. They actively pump carbon into the bunker.
Let's trace the steps of this assembly line. Step 1 occurs out in the mesophyll cells near the surface where the atmospheric air first enters.
The primary CO2 acceptor out here in the open is not RuBP. It is a much smaller 3-carbon molecule called phosphenol pyruvate, or P.
And the enzyme that grabs the CO2 and attaches it to P is called PP carboxylase, or pepcase.
And here is the million-dollar evolutionary advantage. Pepcase is an astonishingly efficient enzyme because it has absolutely no affinity for oxygen whatsoever.
It doesn't care if the cell is flooded with oxygen, it is completely blind to it, will only ever bind to CO2.
So pepcase easily grabs a CO2 molecule and fixes it to the 3-carbon PP, creating a 4-carbon acid.
This first stable product is usually oxaloacetic acid, or OAA. Because the first product has 4 carbons, we call it the C4 pathway.
Okay, so we have safely captured the carbon inside a 4-carbon suitcase. Oxygen couldn't interfere. What is step 2?
The OAA is quickly converted into a slightly more stable 4-carbon compound, typically malic acid or aspartic acid, still sitting out in the mesophyll.
Then comes the critical transport step. These 4-carbon acids are physically pushed through tiny microscopic tunnels called plasmodesmata, crossing the thick cell walls and entering directly into the sealed bundle sheath cells.
The carbon suitcase is successfully smuggled into the bunker. Step 3, decarboxylation. The suitcase is popped open.
Exactly. Once safely inside the bundle sheath, enzymes attack these 4-carbon acids and break them down.
They physically snap off a molecule of CO2 and release it directly into the interior of the bundle sheath cell, leaving a leftover 3-carbon molecule behind.
So they have essentially delivered a highly concentrated, pure dose of CO2 straight into the locked room.
Yes. And what is waiting inside that locked room? Our old, flawed friend RuBisCO, ready to run the normal Calvin cycle.
But the conditions have been artificially altered. Because the Hatch and Slack pathway is constantly, relentlessly pumping CO2 into the sealed bunker, the localized intracellular concentration of CO2 becomes overwhelmingly high.
And because it is absolutely flooded with CO2, RuBisCO's oxygenase activity is completely overwhelmed and suppressed.
The competitive binding is rigged. No oxygen can get in, and there's so much CO2 that RuBisCO functions purely, 100% of the time, as a carboxylase.
Precisely. By locking RuBisCO in a room and pumping it full of pure CO2, C4 plants ensure that photorespiration simply does not occur. Period. They have completely eliminated the wasteful tax.
We should finish the Hatch and Slack loop, though, just to keep the balance sheet accurate. We left a 3-carbon molecule sitting inside the bundle sheath after the CO2 was dropped off.
Right. Step 5 is the return trip. That empty 3-carbon molecule is transported back out through the plasmodesmata into the mesophyll cell.
There, the plant must expend energy, specifically it uses ATP, to convert that 3-carbon molecule back into the original acceptor, PEP.
This completes the cycle, ensuring there is always fresh PEP waiting out in the mesophyll to grab the next incoming CO2.
So as an accountant looking at the final return on investment for the C4 plant, there's a clear cost. They have to spend extra ATP, basically a shipping fee, to run this massive carbon transport mechanism and regenerate PEP. Is that extra energetic cost actually worth it?
Oh, it is overwhelmingly worth it. By spending a little extra energy up front to run this CO2 smuggling pump, C4 plants completely eliminate the catastrophic, debilitating energy drain of photorespiration.
The net result is that they boast significantly higher biomass productivity.
Furthermore, because their pepcase enzyme is so aggressively efficient at pulling in even trace amounts of CO2, C4 plants can afford to keep their stomata partially closed during the hottest parts of the day.
This allows them to conserve massive amounts of water while still photosynthesizing at absolute maximum capacity.
It is the ultimate survival strategy. This is why C4 plants like corn and sugarcane can grow so incredibly fast in brutal, high-temperature, high-light environments where normal C3 plants would simply wither, shut down, and photorespire themselves to death.
It is an absolute triumph of anatomical and biochemical adaptation. The division of labor, the physical separation of the initial carbon capture, and the final Calvin cycle fixation, it is a masterclass in biological engineering.
Which naturally leads us to the final variable in our factory analogy. Even with all this incredible machinery, even with Krantz anatomy and flawless electron transport chains, the factory can only run as fast as its supplies and conditions allow.
You need to look at the market conditions, the factors affecting the overall rate of photosynthesis.
When you look at the fundamental research on this, it all revolves around a critical foundational concept, Blackman's law of limiting factors, which was first proposed way back in 1905.
It is a very logical, almost economic principle. It states that if a complex chemical process is affected by more than one factor, then its overall maximum rate will be entirely determined by the specific factor which is nearest to its minimal value.
The factor in lowest supply controls the entire operation. Think of a real-world assembly line manufacturing cars.
Yeah.
You might have a massive warehouse full of chassis, plenty of engines, and hundreds of workers ready to build.
But if you only have one steering wheel delivered per day, you can only manufacture one single car per day.
The steering wheels are your limiting factor. If you somehow secure a new supplier and increase the number of steering wheels, your overall production goes up instantly until you run out of something else, maybe tires, and then the tires become the new limiting factor.
That makes perfect sense. If you have optimal light, optimal CO2, and a perfectly healthy green leaf, but the temperature suddenly drops near freezing, the plant won't photosynthesize.
All the light in the world won't help if the enzymes are frozen. Temperature becomes the limiting factor. Let's look closely at the key variables Blackman's law applies to in the real world, starting with the most obvious one, light.
Light is obviously the fundamental energy source, but when you look at the data, it behaves somewhat counterintuitively. We have to distinguish between light quality, meaning the wavelengths like the red and blue we discussed, and light intensity.
As the intensity of incident light increases in the morning, the rate of photosynthesis goes up linearly with it. More light equals more power, but it plateaus surprisingly fast.
It hits a hard ceiling. Light saturation actually occurs at just 10% of full direct midday sunlight.
Think about the implications of that. On a bright, clear, cloudless day, the plant is getting bombarded with 10 times more light energy than it can actually process.
Therefore, except for plants growing in deep, permanent shade or stuck down in the dense undergrowth of a forest canopy, light is almost never a limiting factor in nature. They have vastly more than they need.
In fact, too much light is a massive liability. If the incident light increases significantly beyond that saturation point, it literally overwhelms the system.
It causes the physical breakdown of the chlorophyll pigments, a process we called photooxidation earlier, which leads to an immediate, catastrophic decrease in photosynthesis.
The factory basically overheats and burns down.
So if light isn't usually the bottleneck holding the plant back, what is?
By far, the most common bottleneck in the natural world is carbon dioxide. CO2 is the major limiting factor for photosynthesis on a global scale.
The concentration of CO2 in our current atmosphere is remarkably low, hovering between 0.03 and 0.04%.
And here is where the biochemical difference between C3 and C4 plants becomes incredibly visually apparent in the real world. They respond to changes in CO2 concentrations in vastly different ways.
Let's run a hypothetical scenario. Assume light is high enough not to be limiting, and we slowly start increasing the ambient CO2 levels in the air.
Both types of plants will immediately respond by photosynthesizing faster, but only up to a certain point. A C4 plant, because it has that incredibly efficient pepcase enzyme and the internal carbon smuggling pump, will hit its absolute maximum saturation point at a relatively low CO2 concentration of about 360 microliters per liter.
If you pump even more CO2 into the air beyond that 360 mark, the C4 plant doesn't care. It can't grow any faster. Its assembly line is already running at absolute maximum mechanical capacity.
But what about the standard C3 plant, the one stuck relying on the flawed RuBisCO out in the open?
The C3 plant, because it is constantly fighting the inefficiency of photorespiration and the competitive binding of oxygen, requires a significantly higher concentration of CO2 to reach its peak performance.
A C3 plant doesn't reach saturation until the CO2 concentration hits 450 microliters per liter, or even higher.
Because current global atmospheric CO2 levels are slightly below 400, this means that right now, every single day, the C3 plants all around us are actively physically limited by the sheer lack of CO2 in the air. They are starving for carbon.
And human agriculture actually exploits this biological limitation for profit. If you look at high-tech commercial farming, greenhouse crops like tomatoes and bell peppers, which are both C3 plants, are frequently grown in artificially carbon dioxide-enriched atmospheres.
Farmers literally hook up giant tanks and pump pure CO2 gas directly into the sealed greenhouse.
Because CO2 was the primary limiting factor, artificially enriching the air forces the C3 plants to rapidly ramp up their Calvin cycle production, leading to vastly accelerated growth, larger biomass, and significantly higher yields of juicier tomatoes.
It is a massive, real-world application of Blackman's law. You identify the bottleneck and you artificially widen it.
The final two major variables we need to touch on are temperature and water. We already know that the dark reactions, the Calvin cycle occurring in the stroma, are entirely driven by enzymes.
And enzymes are highly sensitive to temperature fluctuations. The light reactions in the membrane are somewhat sensitive, but nowhere near as much as the enzymatic assembly line.
And once again, the C4 plants completely dominate the heat tolerance battle. They are biochemically adapted to respond well to much higher temperatures, showing significantly increased rates of photosynthesis as it gets hotter, while C3 plants have a much lower, cooler temperature optimum before their enzymes start to denature and photorespiration takes over.
As for water, this is an interesting one. Even though water is literally a raw physical reactant in the very first step of the light reactions, remember we meticulously tracked the splitting of 12 molecules of H2O to harvest electrons? Water almost never limits photosynthesis directly by its absence in that specific chemical equation.
Right, because the actual physical volume of water required to supply those electrons is microscopic. If a plant is so catastrophically dehydrated that it lacks the few drops of water needed to run the Z scheme, the plant has already been dead for days.
Precisely. Instead, water stress limits photosynthesis indirectly through its systemic effect on the whole organism.
If water in the soil becomes scarce, the plant's root system detects the deficit and sends an emergency hormonal stress signal up to the leaves.
To conserve whatever internal water it has left, the plant takes immediate evasive action. It firmly closes its stomata.
And as we discussed with the photorespiration disaster, the moment those stomata close, the CO2 supply from the atmosphere is instantly and completely cut off.
The factory doors are locked. The raw materials stop arriving.
Furthermore, prolonged water stress causes the cells to lose turgor pressure. The leaves physically wilt and droop.
This wilting massively reduces the physical surface area of the leaf that is exposed to catch sunlight, slowing down the metabolic activity even further. Water stress causes a systemic shutdown.
Wow. We have covered an immense, staggering amount of ground today. Let's synthesize this entire journey. You have visualized the atomic structure of the chloroplast.
You have tracked the exact quantum flow of light energy, watching photons physically excite electrons out of their orbits in the reaction centers.
You have acted as the ultimate cellular accountant, auditing the precise, meticulous expenditure of exactly 18 ATP and 12 NADPH generated by the Z scheme and chemiosmosis, all just to fund the construction of a single molecule of glucose in the Calvin cycle.
You've navigated the distinct architectural zones of the chloroplast, the membranous power plant generating the currency, and the stromal assembly line spending it.
And you've explored the brilliant, almost devious evolutionary hack of Krantz anatomy, tracking the Hatch and Slack carbon smuggling pathway that C4 plants utilize to completely defeat the crippling energetic tax of photorespiration.
Understanding this level of detail isn't just about memorizing arbitrary numbers for a biology exam, though if you are taking the Neet, visualizing that 18:12 ratio and the C4 loophole will absolutely help you ace it.
It's really about understanding the fundamental foundational economy of the living world. Every single breath of oxygen you take, every single piece of food you ever eat, every calorie of energy you burn is the direct, unbroken result of this microscopic, silent factory running its molecular balance sheet to absolute perfection inside a green leaf.
It is the ultimate basis of the food web and arguably the most successful energy capture system in the universe.
But before we sign off today, I want to leave you with a final provocative thought to mull over, one that builds directly on the quirks we've uncovered in the fundamental biology.
We noted earlier that RuBisCO is universally accepted as the most abundant enzyme on the planet, and yet we just spent a significant amount of time detailing how RuBisCO is actually terribly fundamentally flawed.
It gets easily confused by oxygen. It wastes massive amounts of the plant's hard-earned energy via the photorespiration pathway.
It is so biochemically inefficient that an entire subset of plants had to evolve a highly complex, energy-intensive architectural workaround, the C4 Krantz loophole, just to keep it functioning in hot climates.
So, if we look at this through the lens of deep time, here's the question. Is RuBisCo's sheer, overwhelming abundance on Earth a sign of nature's ultimate triumph, representing an enzyme so vital to survival that life simply brute forces the problem by overproducing it in massive quantities to compensate for its flaws?
Or is its massive abundance just a desperate, incredibly costly evolutionary patch for an ancient billion-year-old mistake? A chemical mistake that perhaps evolved in the primordial oceans long before the Earth's atmosphere even had an oxygen-rich environment to cause the competitive binding problem in the first place.
That is a profoundly beautiful question. It highlights that evolution isn't a forward-planning engineer designing perfect systems. It is a blind tinkerer, constrained by its own history, constantly patching over legacy code.
It is something fascinating to ponder the next time you walk past a tree or look at a blade of grass. You are looking at an ancient, flawed, yet spectacularly successful machine. Thank you so much for joining us on this deep dive into the engine of life. Keep questioning the world around you, keep learning, and we will see you next time.